Hello, and thank you for joining us for the Fawligen and Fall Armyworm Biocontrol Seminar and Accreditation process. My name is Armytage, and I'll be walking through the process today. Looking to run through some things to do with Fawligen, its performance, what we know about the pests, some work in Brazil. We also have Dr. Melina Miles here. Melina is going to contribute some information as well as we're rolling along. We've been successfully able to achieve an emergency use permit for Fawligen. Fawligen was the reason why AgBiTech set up in the US in 2015 to produce the virus for fall armyworm. We're very lucky to be able to establish that operation there, and we produce over 1 million fall armyworm larvae a day to manufacture Fawligen, of which a large proportion of that production goes into Brazil. So it was 1 year, 1 month and 29 days later, after discussions with QDAF, including Melina, that secure an import permit for Fawligen and then secure an emergency use permit. This would have only happened with all the hard work from QDAF. AgBiTech and QDAF have had a very long history of working together. The development of Virus Max came out of some work with QDAF in the early 2000s. Through the process of securing the emergency use permit and the import, we had some written support from PAC Seeds, from Ordco and also from GRDC. So we need to recognise the input from those organisations. Lastly, we do have to mention the role the APVMA fulfilled in this process. They've been nothing short of amazing with regards to the speed and the thoroughness of reviewing the application, securing the data, and turning things around very, very quickly. It's been very, very exciting, and it's been very rewarding to actually have them as part of the team as well. At the end of the meeting, I'll also just point out where there's just a very short little accreditation quiz, which sits on the AgBiTech website. I wanted to run through some population dynamics that relate to these species in the same class that fall armyworm and also Helicoverpa sit in. I think it's important to be able to understand a little bit about the dynamics of how and why, particularly when this pest hit back in January last year, how quickly and how voraciously it spread across the landscape. All these species, these large noctuid moths, they'll produce roughly 1,000 eggs over 10 days of their 14-day life as a moth. This female that we've got here, if you want to run some numbers on this, if of those 1,000 eggs that she lays, if only 16% of those make it through to maturity themselves, half of those are going to be female. So in a generation, which in fall armyworm is about 25 days, particularly in the warmer areas, you end up going from 1 caterpillar to 80 caterpillars in 1 generation. So if you actually spread that out over 100 days, which is the typical lifespan of growing a summer cereal crop, that's 4 generations. So if you multiply 80 by 80 by 80, that one single caterpillar can potentially generate nearly 41 million caterpillars. It's pretty substantial sort of numbers. If you then were to look at what's the situation if you were to be able to halve the numbers of those caterpillars which actually make it through to caterpillars themselves. So from 16% back to 8, that 80 females per generation only drops back to 40 females, which doesn't sound a great deal. But over 4 generations, instead of generating 41 million caterpillars, that takes it back to 2.5 million. This is really reinforcing the whole APM story, and I'm afraid I might be preaching very much to the converted here, but this is relevant when we're actually talking about biological controls, because very few biological controls have the same knockdown or the same amount of efficacy as that you'll find with the mainstream insecticides. But that's a double-edged sword. Now, there are 2 mechanisms for which these species are using to be highly successful in the landscape. And over the millions of years, plants and these caterpillars have evolved together in the landscape. And there's been very much a war of attrition between the 2 of them. As the plants develop new toxins, new compounds, and new defense mechanisms, over the millions of years of coevolution, you find that the pests have also done the same thing. So that machinery relies on an incredibly diverse genetic base. And this is because they're so used to dealing with natural toxins in the environment. And over the millions of years, the tools have got more and more sophisticated as the insects have developed resistance to them. So in actual fact, this has got a lot of relevance to what we do with regards to with insecticide production. So these insects are very used to dealing with a whole heap of toxins which the plants have produced. You'll recognise some of these. Tannin, citronella, gossypol, pyrethrin, they're all natural insecticides that have failed, as are some of these, nicotine, caffeine, and cannabidiol. And particularly these 3 molecules, by some happy coincidence, are psychoactive in a mammalian brain for some other reason. But they are, in actual fact, failed insecticides. For example, one of the biggest pests in medicinal cannabis that get into the grain, that have got extremely high levels of cannabidiol in it, are Helicoverpa caterpillars. So this is the mechanism which these pests have got. So when we come along and we introduce new toxins, these insects are just relying on that same mechanism that they've co-evolved with over millions of years, and they develop resistance very, very quickly to it. Partially because we really put a whole heap of plants together in the same area and then treat the population and apply a lot of pressure. We've also taken some of these natural toxins and made our own versions of them, being synthetic pyrethroids, being spinosyn, and we've also taken toxins from soil-borne bacteria, be the Bt toxin, and again, after a number of a handful of generations, these insects have actually developed resistance to it. This is going to be an ongoing thing because of the pests and that machinery that they've got, the incredibly high fecundity levels and the ability for them to be able to meet a toxic challenge very, very quickly and very, very easily due to the nature of what they've got. However, in that landscape, in that ancient minefield of weaponry, there are some insurgents which operate in that field as well, in the complexity. These are being the beneficial insects who have got a similar reproductive cycle to the pest, and the pathogens. Now, the pathogens themselves, in this particular point, Fawligen being a baculovirus, a virus, has got not a reproductive cycle of 20 days. It's got a matter of several generations in a day. So it can actually beat any of the resistance attempts that the insects do to overcome it. This has got an application because it won't actually take out 100% of the pests, but it will manage the population. So in this landscape, what we've done with regards to Virus Max, which has been in the market now for nearly 20 years in Australia, which is the Helicobacter virus, and Fawligen now being the specific virus for fall armyworm, which is in the picture here, we then have weaponized it. So how do these MPVs work? How do these viruses work in here? And this is important. Again, apologies for those people who have been using this product for a long time, but effectively the caterpillar ingests the virus particle, which is embedded in this inclusion body, and then it goes into the digestive tract. In the lower digestive tract, the virus in the inclusion body dissolves, the virus is then released and it binds to the gut wall. From there, it then spreads all the way through the caterpillar and hijacks the repair mechanisms which actually sit within the DNA of the insects and spreads through the insects. It then liquefies the insect dies, and that so then releases a very large volume of virus into the environment. Now, we'll come back to that. Putting this into perspective, Fawligen contains 7.5 billion occlusion bodies per mil. So it's a massive numbers game. Virus Max, for example, contains 5 billion occlusion bodies per mil. And the occlusion body itself is just a small plasticine sort of particle that's actually got a lot of the virus packed in it. So we're dealing with incredibly big numbers. That is the mechanism by which these viruses all survive in the environment. Now, I'd just like to move to the Brazilian experience, and keeping in mind that the reason why AgBiTech set up in the US in 2015 was to be able to supply the Brazilian market largely and the US market with baculoviruses to control insects, specifically the fall armyworm. If you look at Australia, and I just draw your attention to the green areas here. So the green areas are very obvious. They're in the north, the east, and also the southwest there. But then if you actually look at this in comparison to Brazil, you actually get to see this green belt, this large green belt here. And this is where fall armyworm as a pest has evolved. And this is its natural environment. So if you were to overlay Brazil over the top of Australia, or move Australia to where Brazil is, effectively you would have a green bridge that runs from Perth to Brisbane as a lush, green, highly alive area. So this is the environment the fall armyworm have come out of. So when you look at it on a global map, you can actually see here that here's the top end of Brazil, Ecuador, Colombia. This is where this pest comes from. And the reasons why it's called fall armyworm, because in the autumn or the fall, it will migrate northwards as far as the Canadian border or south to the middle of Argentina. So if you were to look at that across as an analogue here in Australia, you would think that southern New South Wales, being in the autumn, we should be seeing fall armyworm no earlier than perhaps March or April in the southern parts of New South Wales or into Victoria. But in actual fact, the case was it was in November that we picked it up in southern New South Wales. So this pest entered through the north, and within a matter of 10 months, it actually put itself down to the bottom, of New South Wales. Now, the other piece of information here is that AgBiTech in 2015 were also requested to send a large volume of Virus Max to Brazil because Helicobacter armyworm had broken out over there. And what happened after about 18 months, the population crashed and we got a call to saying the pest had largely disappeared from the landscape. Now, I don't want for a moment to suggest that this is going to be the case with fall armyworm. But what I would like to point out to you is that the pest now in Brazil, being Helicobacter pyometra, is considered to be a secondary pest. So what I'm suggesting here is that the first couple of years of the introduction of an exotic pest into a landscape, it will spread and explode across the landscape largely unhindered. And what tends to happen after a couple of years, that the pest will then settle down into some sort of natural balance within the environment. The other thing too, which Melina Miles from QDAF has also pointed out, is that this year just past has been almost a perfect storm as far as armyworm generally. So we've had a very good season for armyworm generally, the common armyworm, for example, and then we've also had the mummy daddy of all armyworm invade from the north as well. So there's a bit of a double whammy there. So the take home for this is that we're dealing with a pest in the north, we're dealing with a pest in the south, but in the next couple of years, things are going to change, but we don't know what it is. So we're going to have to very heavily rely on the scientists and the science to understand how this pest will settle into the landscape as we go further along. The other thing too is scouting for this pest can be a problem. In those intensive row crops where we've got lots of experience of scouting for Helicobacter eggs are scattered through the canopy. So 10 eggs to the metre are relatively easy to find because they're scattered right through the crop. Whereas 10 eggs to the metre, given the fact that the egg cluster from fall armyworm, there can be 10 metres worth of eggs in one egg sac. So it's going to be very, very hard to scout for it. This has got implications for management of it because you can go from effectively having a clean crop to having a lot of the pest in the crop in a very short period of time. We've also, AgBiTech, we've been working in Africa and South Asia as well. And here's some photos out of Nigeria and also out of South Sudan of the pest that's there. Now, what we also know is that the pest behaves differently. The pests in Brazil is behaving differently with regards to resistance, with regards to susceptibility to Fawligen as compared to Africa. And we're still trying to unpack the reasons why for that. There, again, we need some science. But it is shown the fact that the pest we have here has migrated to Australia via China, via India. It's also then come down through Indonesia, and then it's coming to the north from there. So the work that's done by CSIRO, looked by QDAF, New South Wales DPI, are really looking at these populations to see how it is. But the early bioassay work that's been done here in Australia is showing that the pest that we have here is more susceptible to Fawligen than we're actually seeing to the pest in Brazil, which is very good news for us. In talking to our team in Brazil, we've got 70 people on the ground in Brazil and we've got 40 people in the US. We're talking about host crops and fall armyworm. And it's come through very clearly. From a host crop perspective, this pest is a pest of maize or very close relatives. Maize, popcorn and sweet corn are its absolute delicacies. But when it gets into large numbers, it will move into these other crops. And we are seeing this, and Melina, I'm sure will speak to this, we are seeing the fact that we're seeing this pest in other crops, but it is nowhere near as damaging as we're physically seeing in the maize. So this is one of the things that we need to understand a little bit, is that what is the relationship with this pest in these other crops? And again, we only have the experience that we have in Brazil and Africa, which would indicate the fact that it's primarily a pest of maize. And what we're seeing so far is it's similar in Australia. I went to great lengths with our team in Brazil to try and get them to explain to me what was the relationship of fall armyworm and sorghum, grain sorghum, for example. And it did take a lot of time, but eventually I got some photos through of, and this was 8 months ago, of fall armyworm in sorghum. Yes, it gets into it. Yes, it will get into the head like Healy's will, but it seems to be nowhere near as a bigger problem as Healy's can be, unless it's in very, very large numbers. At this point in time, Melina, I might just even hand over to you on this, just to run through some of the stuff that you're doing in the broader picture.
Sure. Thanks very much, Philip. So I guess what Philip's asked me to do is just to talk a little bit about, the key difference between what we've been doing in terms of caterpillar pest management and fall armyworm. And for those of you who've had experience with fall armyworm, I think that the overriding experience has been that this is not a simple animal to control. I guess the approach that we've always used with caterpillars is to check to assess whether the density is above or below threshold. If it's above threshold, apply an insecticide and the problem is resolved. That is definitely not the experience to date with fall armyworm. Getting good levels of control is really challenging. And the other thing that's really challenged many growers and their advisors has been the speed with which fall armyworm establishes and causes quite significant damage. goes through its life cycle, as Philip said, quite rapidly, and particularly those early instars that get themselves into positions in the crop, in the whirl and so on, where they are much more challenging to contact with insecticides. So what this slide really describes is, those coloured boxes, which is the approach that we've used to date with caterpillars, to focus on not necessarily looking for eggs other than an indicator that there's some activity, but really counting the caterpillars and working out if we need to control or not. What I am suggesting, we need to start thinking about with fall armyworm is population suppression. That at every opportunity we need to be thinking about how we suppress that population in the crop to minimise the number of large larvae that are there causing significant crop loss, both in the vegetative, at establishment principally, and then during the vegetative stages and subsequently the reproductive stages. So if we start on the left-hand side where you can see the picture of the moth, we're quite fortunate that we do have some options in relation to suppressing numbers of moths that go on to lay eggs in the crop. So magnet is one of those, an attract and kill proposition where it's applied to the crop to kill moths before they lay either any eggs or their full complement of eggs. There are other products that are available overseas, not yet in Australia, that do a similar thing through a mating disruption approach where you spray out a pheromone that confuses the moths, means that the males and females can't find each other effectively, and you get much fewer viable egg masses laid. Things like intercropping, trap cropping, and then just mass trapping with things like light traps have also been discussed as options, but those, I guess, are much less developed than those others that I mentioned just before. Then, of course, there are the eggs, and to my mind, targeting eggs is, or facilitating mortality of eggs by the retention of natural enemies, either predators or parasitoids, is absolutely a terrific place to put some effort, because if you don't get larvae emerging from eggs, you're really in a much better position than if you allow that to happen. So we already know from the sampling that we've been doing across the Downs and up into North Queensland, that there's a good complement of natural enemies that are attacking fall armyworm. And as far as eggs go, trichogramma, whether that's pretiosum, the introduced and commercially available one, or native trichogramma, is unclear. We're in the process of having those identified. But there is real potential, if you can retain natural enemies in the crop, to affect a really good population suppression. The other part, and I guess, from a Helicoverpa management point of view, we're a little bit familiar with pupae busting. It's a little bit more sort of complicated with fall armyworm, because you can get many generations, or at least 2 or 3 generations per crop. So the opportunities to pupae bust are somewhat limited, but there are options with things like pathogens, so either fungi or bacteria or even nematodes, which are things that we haven't really needed to give any attention to up till now. So I think what I wanted to do is just to sort of, broaden that perspective, whilst Fawligen is something that is targeted at the caterpillar stage, that, the benefits that it brings in terms of providing a good environment for natural enemies to also attack these other stages is an important part of the conversation. Do we move to the next one, Philip? So just to give you an idea, most of these photographs, apart from the trichogramma ones, are from Australia and things that we've already seen in the field, as I mentioned. You'll be familiar with many of these because they also attack other caterpillar pests like the native armyworms and Helicoverpa. So, you'd be familiar on the bottom left there with Cotesia that leaves little white clusters of cocoons. It really, in northern New South Wales, was almost solely responsible for halting populations of fall armyworm back in September, October last year. Predatory shield bugs, absolutely voracious, particularly the spine predatory shield bug. It turns up sort of in enormous numbers into crops and seems to be quite an effective predator on fall armyworm. And then given that it was a wetter year this year, we've also seen epizootics or natural outbreaks of things like Metarhizium riley, the fungal pathogen that you can see there on the top right. And DAF has some considerable interest in pathogens, and Ian Newton, based up in Mareeba, is starting to do some work both on the naturally occurring viruses and also on this Metarhizium riley. So there's real potential, I think, to bring together the different pieces that will be essential to get good control of fall armyworm. Next one. Okay, so Philip mentioned that we've started the process of looking at how Fawligen interacts with the fall armyworm population that we have here in Australia, and I guess not making any assumptions about how it will respond in comparison with experiences elsewhere around the world. So when Vivus was launched in Australia, there had been sort of 20 odd years of work by DAF, by Bob Tickle, on, what were the components that were important in deploying a virus in the field. We unfortunately don't have that, that information for Fawligen or for NPVs of Spodoptera frigiperta. So we really are starting from first principles. And part of that is just understanding what the relative susceptibility is of the different life stages. And that is fundamental to how you target a population, particularly a population like fall armyworm, where it's highly probable that you're going to have a range of larval sizes in the crop. This is one of the very first bits of work that we've done with basically just working our way through the different instars. So that graph shows each one of those coloured lines is a different instar from first through to sixth. And the really important thing to see here is that the efficacy of Fawligen is quite good. Equivalent to the sort of efficacy that we see with Vivus against Helicobacter armyworm for first and second instars. And then there's an enormous drop-off in efficacy down to third instar. And by the time we've got to fifth and sixth instars, there's virtually no effective control of those, no effective infection by Fawligen. So this is at 200 mils. I guess, some of the work that we're doing now is looking at what dose it will take to pick up those thirds and perhaps fourths, and also whether there's benefits in including optimal, or what benefit optimal may provide, not only in terms of infectivity of larger larvae, but also whether it promotes the longevity of the virus in the crop so that you might get more larvae ingesting it, and various things like that. So that work's ongoing, and we've got a number of bioassays running at the moment looking at some of those questions. Okay, so these are just a few photographs that I've been taking as we've been doing bioassays in the lab, and I guess those two on the bottom right, or the middle and the bottom right, probably look quite familiar if you have some experience with Helicobacter. That's fairly typical for, sort of, I guess, the progress of virus infection and when the larvae start to disintegrate and release the virus. We have noticed already some quite significant differences in the behaviour of larvae infected with Fawligen, so Spodoptera larvae. And one of those is that we're not seeing the larvae come up to the top of the cups like we tend to see pretty reliably with Helicobacter. And we're not, it's not necessarily resulting in all the larvae oozing like those two at the bottom right. On the bottom left, you can see one that's sort of oozing a dark, noxious looking fluid, and same top right there. That's also something that we're seeing quite often. The only other consistency that I could describe from what we've seen so far is that picture that's up there on the top left, where the larva is quite pale, clearly, not healthy looking. And that seems to be the case with many larvae. And they don't necessarily progress to that oozing stage. So there are some differences. And I guess, we haven't done a lot of work in the field yet. So I don't know what it looks like in the field, consistently, but I suspect that you will struggle to find larvae that are showing typical virus symptoms, particularly, because they don't seem to move to the top of the plant where they're easily observable. And they also are quite small, first and second larvae challenging to find at the best of times. I guess this is a work in progress to help you to understand how best to make an assessment of what the efficacy of Fawligen is on fall armyworm.
More specifically around what we know about the performance of Fawligen versus Vivis Max. So we have a very long use of Vivis Max and the new formulation Vivis Armytage in Australia that goes back almost 20 years. In fact, in the last couple of years, more than 80% of the sorghum in Australia has been sprayed with Vivis Max. So there's quite a large experience base there in Australia. Fawligen is obviously the same virus, but it's very specific to Spodoptera frugiperta for fall armyworm, and it's got no impact at all on Helicoverpa. However, there are some significant differences with regards to the performance. As Melina's just showed us there, the susceptibility of the larvae is mainly first and second instar larvae. So if we're looking at actually comparing larvae sizes of use window, in Helicoverpa, we do know that you can pick up the larger larvae, although it's not recommended. And these larger larvae, we suspect, are very much the reasons for the epizootic effects which roll out into the environment. But with Fawligen, it seems to be only those first and second instar larvae that we can reasonably reliably pick up if we get the caterpillar to physically ingest it. Talking about application rates. So be aware of the fact that because we have an emergency use permit for the product which is unregistered in Australia, there isn't a huge body of work in Australia which actually would confirm the use rates. So we are working off the experience that we've got from the US, from Brazil, and also through Africa with regards to the use of it. The application rates for cereal grains run from 50 to 200 mils plus optimal, and I will come back to the optimal adjuvant in a second. The data we have from Brazil is suggesting the fact that the dose response is quite flat from 50 to 200 mils. Again, Melina's running the bioassay work at the moment to see if we can actually confirm this. So if we're talking cereal grains at the moment, we're talking maize. For want of a better number, we're recommending we look at probably 100 mils to the hectare, and we actually look at it, putting it on a band going onto small maize. If you look then at sweet corn, the sweet corn rate is 200 mils to the hectare. And then if we're looking at oilseeds and pulses, which we're not really believing we're going to have too much of a problem with this pest in, it's the same application rate with regards to cereal grains as in maize. Timing, and this is still very much a work in progress. Because we know it's not going to work on the larger larvae, our recommendation is to go early season and work pre-threshold work on a band spray. Part of this is that from a cost perspective, we're looking at the 100 mil rate per hectare. We're looking at only $20 a hectare. So on a 50% band, that's starting to look quite cost effective. But that's in recognition of the fact that it is not going to pick up any of the larger larvae. So we would strongly suggest look at using this early season, almost on a program basis, to try and shut down those early numbers of smaller larvae as they're coming through. We're also recommending that because there are no resistance to this product whatsoever, if the caterpillars ingest it as first or second instar, it will kill them, that it will be a very good mixing partner for later season, particularly when you're using the more expensive chemistries to help with regards to the smaller larvae and also from a resistance perspective. The other thing that we've noted also is that compared to Vivis Max on Helicoverpa, the time to kill is slower. It can be by 1 or 2 days. We're not exactly sure why that's the case, but it seems to be the case. Because it's only working on first and second instar larvae, it's very hard to find the dead larvae. And as Melina said, from the behavioural perspective, we're not seeing the bigger caterpillars climb to the top of the plant and dying. We're finding the smaller ones are dying and falling off the plant. So this has got implications for the epizootic stuff, but I'll come back to that in a second. The implications for the epizootic effect, as in the rolling infection, because I think there's just so little biomass with the smaller caterpillars getting control with it, they're not generating a huge volume of virus. So we're unlikely to see epizootic effects in the field. However, as this is the means by which it spreads in the environment, we know it has to work to a degree because this is how this particular pathogen exists. Some other key information, and just coming back to this, coverage, and more coverage. Minimum in maize crops, minimum of 100 litres per hectare, 400 litres per hectare is good. A band spray, humid conditions are good, and the breakdown of the virus is by UV light. So if you actually look at the image here we're using as the backdrop, it's not a very friendly environment for a product that gets broken down by UV light. The product gets applied to the leaf blades and After about 2 to 3 days, it's largely broken down by the UV light. However, if you can actually get it down into the whirl and get it into the protected spaces, it will last for a great deal longer, which I'll come back to that because there's also an issue, an idea with regards to the water running aspects of it. Now, we're recommending the use of Optimal, which is recommended for use with Vivus Max and Vivus Armyworm. Now, what Optimal is, it's a spray oil and it's got high fructose sugars and sugar syrup with it. There was a lot of work done which actually demonstrated that Optimal can and reliably does improve the infectiveness, the infectiousness of Vivus Max. We don't have any work from the US or Brazil on this front. They're not up to that stage yet. So but we are recommending that we use it because we believe it will actually assist with the infection rates quite clearly. The use rates for that is 1 litre per hectare. If you're using 100 litres of water to the hectare, it's 1 litre. And above that, put in 2 litres per hectare of Optimal. We believe that we can get a better result with it. Secondly, then, is the time of the day. So we know from our work with Vivus Max and Helicoverpa, 90% of your infection occurs in that 1 hour after application. So Helicoverpa can be quite day active as far as larvae goes, whereas we know Fall Armyworm are quite sensitive to light. Particularly the larger larvae tend not to be day active at all. The smaller ones can be, and we're getting some feedback from the field that would very much be the case. So whilst we're not being too prescriptive about this, be aware of the fact that if we're looking to get that infection, it really is, you do need to target the time of application when the larvae are most active on the plant, not so much when they're fully entrenched and hiding either from the heat of the day or the cold of the day. As with Vivus Max, rain fastness, a light shower or heavy dews actually work in your favour for actually spreading the product around and getting it down to the whorls. Heavier rain will wash it off the plant. So it is not rain fast at all, but it will, under light conditions, light showers and heavy dews will actually assist you with it. The gut of these pests is different to our gut. The gut of these pests is actually an alkaline gut as compared to an acid gut that we have. So as a result, those occlusion bodies are designed to dissolve in an alkaline environment in the gut. So our recommendation, as it is for Vivus Max, is to ensure you've got a pH of 7 water. If you're getting a pH of 8 or above, make sure that you are using a buffer to try and manage that pH down. But it is quite critical to make sure that we give the virus the best opportunity possible by using the best quality water that you physically can. Some work that's also been done, again by Melina and the crew, has demonstrated the fact that there's no antagonism by mixing Vivus Max or Vivus Armyworm with Fawligen. There's no antagonism either with the Heelys or with the Fall Armyworm, either which way. So it is possible to be able to mix the two if required and have confidence that you're not going to get an antagonism. Keeping in mind that the particular season, you will end up with a mixed population with Heelys. It would seem to be that the small maize in particular is the most attractive to Fall Armyworm. The later season, where they're starting to attack the cobs, you will start to find a mixed population with Heelys. One thing that I'm recommending is that prior to using the Fawligen, make sure you decontaminate your equipment. Now, it's a bit of a no-brainer if you've been using herbicides, but I'm recommending here decontaminate if you've been using any other insecticide. For example, Spinetoram Success has a very soft profile as far as a lot of the beneficial insects go, but it is very, very hard on trichogramma. The same as Should is very, very soft on many species, but it's very hard on your beetles. So what we would suggest to give the product the best chance of working is make sure you decontaminate your boom to take those residues out. Synthetic pyrethroids, methanol, they're all very hard on these beneficial insects. And half the work that we're doing is being done by the virus killing the caterpillars, and the other half is by the beneficial insects doing the work for you as well. Having said this, also, the product is very, very compatible, as is Vivus Max, with almost every other insecticide. We really don't see any problems with any compatibilities from a mixing perspective. Because being a biological product, re-entry is when the material is dry in the plant, and there is no withholding period when the product's used as per the label, which is attached to the emergency use permit, which is good news. I would like to touch now on water running Fawligen. We know water running Vivus works incredibly well, particularly later season in maize for Heelys. And we did a little bit of work as to trying to work out why this was the case. And If you remember back to the numbers, if there's 7.5 billion occlusion bodies per mil, and you've got an irrigation system which is applying a 10 millimetre watering event, that's an ideal situation for actually water running it. So if you were to look at just conventional dye through a boom application or by an aircraft, here's some work which I do with some maize, and you can actually see the coverage that you're physically getting. More particularly if you actually zoom in on the end here on the silks, you can actually see that there is some insecticide which actually gets targeted onto the silks. The rest of the cob then is largely unprotected. And one of the things I was trying to do was trying to work out why do you get such a good job with Vivis Max when you water run it chasing Heelys, particularly the Heelys that get into the end of the cob. This is the same thing when we actually water run it with the dye. The dye doesn't tend to stick terribly well to the kernels of the maize, but you'll find that the silks actually wick water right the way down through the cob, all the way through it, so it's getting it in. So if you're then looking at actually the end of the cob where the Heelys get into in the end of the cob, it's very easy to target them. So this is what we're suggesting with Fawligen, is that although we don't have the data for it, I would strongly suggest that we can get a similar type attack on these pests when they actually target in the side of the cob, because you can actually get some very dilute, but you can actually get some of the virus actually to where the insect is physically feeding, where it's physically entrenched. If you're water running the Fawligen, you don't need to use optimal. The optimal is only for use if you run it by boom or by aircraft. I'd now like to talk briefly with regards to storage of Fawligen. Fawligen needs to be stored at 4 degrees or lower, and as a biological, this protects the breakdown of the product. It can be stored at room temperature of up to 27 degrees for less than a week, but that is not ideal. It's also very sensitive to UV light, so it also needs to be kept in the dark. It can also be stored in a domestic freezer, which goes down to -18 and -20 degrees, as the product won't freeze and it doesn't hurt it at all. Long-term storage, this is the ideal way of storing it. Critical summary here, if you take away anything from this 45 minutes, only use on first and second instar larvae, use it sub-threshold. Make sure complete coverage is critical for getting that application. If you can move to a band spray by turning your nozzles and actually getting a 50% band or smaller, put the additional water down the plant and onto the plant to try and get the coverage. Use it as a pre-threshold or as a mixing partner. Be very wary of trying to use it as a knockdown in its own right. If you get those first and second instar larvae to ingest the virus, you will kill them, but you've got to get them to physically ingest it. And the last one here, the whole reason why we're actually doing this part of this program is to assist everybody with managing the performance expectations around the product. It's not as effective as Vivis Max is on Heelys, but on the other hand, it is another tool that we can physically put into the system. The other part of actually the reason why we're running today is that one of the constraints the APVMA have put on us with regards to the emergency use permit is that all the users are recorded and the uses of it's recorded and available for the APVMA. So we're going to be working with our distribution partners to be able to source those people that have actually acquired the product to make sure that we can get access to, one, that they've purchased it, and two, that if we need to, we can reach through them to find out when it was physically applied. It's probably quite a long shot, but I would recommend to you that if we can have that data available, it will make it very easy should the APVMA request it. I just want to come back to the fact again, the APVMA have been absolutely incredible with this process, with actually getting this tool through to you growers. After we've finished here, on the Fawligen page on our website, there's just a little quick survey which will then capture just to ask a couple of questions on some things that we've already covered off. So if I would encourage you just to be able to do that, and that'll then give us a record of those people who have been through this process. Finally, just touching on the supply status, we've flown in a volume of product in 2 tranches with the support of QDAF. We now have that bottled and we now have that ready for use now. So that's happened. So again, that's all happening with 1 year, 1 month and 29 days work. So it's been quite a phenomenal effort by distribution, by the APVMA, as well as also the federal agencies on importing a virus into Australia. So with that, I'll just open the floor now for questions. Please feel free to message those through, or if you want to unmute and actually bring them to the table. There's a question here I see with regards to the number of applications. That is actually sitting on the label, which I can track it down, but it is substantial. I think we're allowed up to 10 applications on maize. I think it is, but that information is available on the label, which is on our website as well, but substantial. One of the other things that just come in here with regards to storage of it, it is the same as Virus Max with regards to storage. So it can, it requires for long-term storage, it needs to be stored at 4 degrees or below, which is a conventional refrigerator. It can be stored in the freezer. However, for shorter periods of time, a matter of days, even a couple of weeks, it can be stored at 25 degrees C, which would allow it to be stored in an office or within an air-conditioned house as required. Once you get over that temperature, the breakdown of the product happens very quickly. It's not like ice cream. Once it's warmed up, it's had it. It's not that case, but it needs to be stored at 4 degrees or below. When we transport it any distances, it's transported in refrigerated conditions. But if it's going out to an airstrip, it's going onto a farm. As long as it doesn't get over that 25 degrees C, the product will be perfectly fine. The same as it is for Virus Max. I've just got another question coming here from Monica. Can you elaborate on the time of the day, early morning? Again, we just need to understand about the pest and its behaviour. And Melina, I wonder, with your experience with the trial work you've been doing, could you shed a little bit of light on the behaviour of the insect when it's likely to be most active?
Well, I guess there is some reasonable observational data to suggest that the larvae are much more active at night. You know, when we have sampled trials during the day and then come back at night to have a look at magnet, I guess, have been really surprised at the number of larvae that are apparent in the crop that weren't there when we sampled during the day. So to some extent, they're coming out of the ground, particularly under high density. You'll find that there's a fair proportion of the larvae will sit under the surface of the soil during the day to avoid the heat and the sunlight. They're quite averse to sitting out in the sun during the day. So I guess there are two parts to that question. One is you want to be getting the virus on, the Fawligen on, pretty close to the time that those larvae will be active and feeding. So that's both probably for fall armyworm, that's more likely to be overnight. But if you're in an area where nighttime temperatures are sort of dropping down below 18 degrees, then perhaps that, that will constrain their activity a little bit. So yeah, I think, from what we've seen and I guess what we know from the behaviour so far, if you can put it on late in the afternoon, so it's there when they become active before it starts to cool down too much, that at the moment seems to be the best strategy.
Thanks, Melina. Another question here with regards to seed treatments. There is work being done. I think actually Syngenta have been doing work with the seed treatment here in Australia, which is coming through registration process. I do believe that actually looks quite encouraging. Again, anything we can do to preserve those early season beneficials will assist us with getting it through. And I think a seed treatment is ideal because it's then contained inside the plant, even if it is a little bit reasonably toxic or anything feeding on the plant. It then allows us then, if you were using Fawligen and the highly selective products, to get through from V2 through to V10, which really seems to be with the critical window. The other question I've got here, a direct question, is with regards to magnet use. So the questions we regard, what is magnet? Magnet was developed by the cotton industry in the early 2000s and was registered in 2008. In effect, it's a moth attract and kill. So it's a thick, syrupy product that's got volatiles in it, which the moths are very, very attracted to. It's laced with an insecticide and then applied in strips through the crop at either 36 or 72 metres. And the moths, over about 2 to 3 days, the moths which are nearby will find this, feed on it, and it will actually control them very, very quickly. Again, it's very good for use under light to moderate pressure, but under heavy pressure, it's not likely to be terribly useful because it becomes a numbers game. But it can take out, if we used on a large scale, it can take out up to 80 or 90% of the moths when you're using it over several hundred hectares. It doesn't work if you're trying to use it on a few hectares because it's akin to going to the beach with a shovel and trying to dig a hole in the dry sand. The moths are just pouring in from the outside. But it again is a useful tool. There is currently a large volume of that about to go out up in the Ord over this next 3 to 4 weeks, using it in conjunction with Fawligen to try and manage this early fall armyworm population while the crop gets away from this critical stage, which is V2 to V10. that's 45 minutes, everybody. So look, I don't have any more questions coming on the chat line. Please feel free to reach out to us through Ian or myself. Melina, I know, is always on tap as far as the industry support goes. We do have stock available for those people that want to be using it now. And if you have a chance just to run through that short little accreditation quiz, that'll also then get you in the system with that. Thank you very much, everybody.